HIV is a retrovirus that is primarily infecting the CD4 positive helper T cells. So there are surface proteins that are called GP120, and GP120 will bind to the CD4 receptor, after which it causes conformational changes and make the CCR5 and CXCR4 co-receptors available for binding. So next, GP120 will bind to these co-receptors, and then after that, the GP41 protein will induce the infusion of virus into the cells. So individuals that have mutation in CCR5, if they have homozygous mutations, then they're immune to HIV infection. And if they're heterozygous, then the course of the disease would be very slow in these individuals. So mutations in CCR5 co-receptors make patients either immune or resistant to HIV infections. Now here there are genes that will express the HIV proteins. The ENV gene will express the GP120 and GP41 protein, which we've just discussed their functions. Then there is a GAG gene which produces the caspase proteins. And then finally, POL will produce 3 different enzymes, which include the reverse transcriptase. So what happens is that HIV is an RNA virus. So RNA will be reverse transcribed to DNA, and then later on by integrase enzyme, this DNA will be integrated into the host genome. And then this DNA, via transcription, can produce mRNA, and then from mRNA, polypeptides will be formed. And from here on, the protease enzyme helps break down the polypeptide to smaller functional proteins. So there are different tests that you can use for diagnosing HIV infection, and these include the ELISA test, which is a sensitive test. And if someone is testing negative for ELISA, then you can rule out the disease. However, if the patient is positive for ELISA, then you will have to use the Western blot next, which is a highly specific test. So if the patient now tests positive for Western blot, then you will rule in the disease. Next, I would like to go over the infections that are more commonly seen in HIV patients. So here I have the clinical findings and the lab findings, and then we would like to know what is the infectious agent. So if the patient presents with fever, cough, tongue ulcers, and hepatosplenomegaly, and then the lab findings shows yeast within the macrophages, then it's a histoplasma infection. So here is an example of a macrophage, and then there are yeasts that are inside the macrophage. So these are all the histoplasma that are engulfed by the macrophage. The next condition is if an HIV patient presents with a chronic watery diarrhea and examination of the stool shows acid-fast cyst, then this is an example of a Cryptosporidium parvum. So I would like to remind you that the organisms that will test positive for acid-fast include the Mycobacterium nocardia as well as the cyst of the Cryptosporidium parvum. The next 2 conditions are the vascular proliferation. So if on biopsy you see lymphocytic inflammation, then that should make you think of Kaposi's sarcoma, which is caused by human herpes virus 8. And then if you see neutrophilic inflammation, then that should make you think of the cat scratch disease, which is caused by the Bartonella hen cell. And so here is an example of Kaposi's sarcoma. So you can see the vascular proliferation, and if you do the biopsy, you will see lymphocytic infiltration. And then here is an example of a cat scratch disease where there are plenty of neutrophils here, and it's important to note that they have a stellate shape of neutrophils that are presenting right here. Next, we have 2 conditions that present with ring-enhancing lesions. So both this one, the CNS lymphoma, and the brain abscesses that will present with ring-enhancing lesion. And the first one, CNS lymphoma, is being caused by Epstein-Barr virus. And then the brain abscess, which presents with multiple ring-enhancing lesion, is caused by toxoplasma, and usually in patients that have a CD4 count of less than 100. So one giveaway point to differentiate between these 2, because both are presenting with ring-enhancing lesions, is that the CNS lymphoma has focal lesions, while brain abscesses have multiple lesions there. So here is an example of the ring-enhancing lesions. So if I ask you, is it a toxoplasma or a lymphoma, since there are 2 lesions that are shown here, I would go with toxoplasma for this example. The next disorder is meningitis in HIV patient, and there is a narrow-based budding yeast, and this is caused by the Cryptococcus neoformans. And this is a heavily encapsulated yeast, so here you can see that there is a heavily encapsulated, so the capsule is really thick that is around the yeast right here. And then you will use India ink to stain the Cryptococcus neoformans. And then this yeast is more commonly found in pigeons' droppings. The next condition is retinitis, and then these patients present with cotton wool spots on the fundoscopic examination. So that would be an example of CMV retinitis. And here is an image. So here you can see the cotton wool spot on the image of the retina, and this is an example of CMV retinitis. Next, we have dementia, and this can be caused by HIV virus itself. Encephalopathy with demyelination is being caused by the JC virus. Squamous cell carcinoma around the anus is being caused by Epstein-Barr virus, and Epstein-Barr virus can also cause hairy leukoplakia, which usually presents on the lateral tongue. So this is another example of Epstein-Barr virus infection. Now, if you have fluffy white cottage cheese lesions, then that's an example of infection with Candida. And Candida can present in multiple different forms, which include the pseudohyphae and germ tubes. So here are different forms of yeast that can be shown. So it can present as a budding yeast, it can be as a true hyphae, pseudohyphae, or germ tube. So if you see either the pseudohyphae or germ tube on exams, you should right away think of Candida albicans infection. Next, we have interstitial pneumonia with intranuclear inclusion bodies that have owl's eye presentation, and this is another example of CMV infection. So CMV can both cause the retinitis as well as interstitial pneumonia in these patients. And here is an example of the owl's eye. So here you can see that there is a nucleus with an intranuclear inclusion that has a presentation of owl's eye. Next, we have a patient, HIV patient with pleuritic pain and hemoptysis. An examination shows fungus ball, and this usually forms in the preformed lung cavities. And this is an example of aspergillus. And here is a drawing of aspergillus, so you can see that it has septated hyphae at 45-degree angles. Next, we have pneumonia in patients that have less CD4 count of less than 200, and this is a high-yield topic because it's being caused by the Pneumocystis jirovecii, and it's usually seen in patients with CD4 count of less than 200. On the other hand, if they present with tuberculosis-like syndrome in patients that have CD4 count of less than 50, then you should think of the Mycobacterium avium. So there are prophylactic treatments for these infections, and so for Pneumocystis jirovecii, so the other name for it is Pneumocystis pneumonia, or PCP, you will have to use sulfamethoxide trimethoprim. And for patients that have CD4 count of less than 50, then you have to also add the azithromycin to protect against Mycobacterium avium. Now, I would also like to mention that the sulfamethoxide trimethoprim can also be used for the treatment of the toxoplasma. So if you recall, toxoplasma can cause ring-enhancing lesion if the CD4 count is less than 100. So the same sulfamethoxide trimethoprim that is used as a prophylactic treatment against PCP can be used for prophylactic treatment for toxoplasma. Now, if a patient is allergic to sulfamethoxide trimethoprim for the treatment against pneumonia, you will have to use either dapsone or atovaquone. An alternative option is pentamidine, but it's not as efficient as the other two. So this sulfamethoxide trimethoprim is by far more efficient than the other medications that I've listed here. But if the patient is allergic, then these would be the alternative options that we can use. And one last point that I would like to mention is that I earlier mentioned that Kaposi sarcoma, which is caused by the human herpes virus 8, is an AIDS-defining illness. The other two cancers that are AIDS-defining cancers include non-Hodgkin lymphoma, and then finally we have the invasive cervical cancer. So these three are considered as AIDS-defining cancers. And that concludes our discussion.